Hello everyone. Today we are going to do one very important practical which is used in the field of molecular biology and RDT, that is the polymerase chain reaction. Now we all know that this polymerase chain reaction is a technique by which we can enzymatically replicate the DNA without the help of any living system. This process was invented by Professor Carey Mullis, and for this invention he was elected and he got Nobel Prize for it. So just let me discuss what are the things that we need to set up one PCR reaction. So first, there are several things that we need. One is basically the template DNA. Second is we need the primer. So the primer that will go and bind specifically to the site in the DNA molecule. So there are two primers we need. One is the forward primer, another one is the reverse primer. So that's why two primer I have written here. Third most important thing, we need to supply the nucleotides, that is A, T, C and G. Fourth one, as I mentioned, that is an enzymatic process for the replication. So that's why we need to supply one very important enzyme that is isolated from Thermus aquaticus. It's a Taq polymerase enzyme. Fifth one, we need a suitable buffer so that this enzyme have a maximum activity using the buffer. Next one, we need a small tube. We call this a PCR tube. So PCR tube has got a very small volume capacity. And finally, we need that a nucleus-free water. Okay, so now how to set up one experiment with all of this component I am going to discuss later.
Like thin wall PCR tubes, ice bucket, agarose gel, and then forward primer, reverse primer, DNTPs, template DNA, Taq enzyme, specific buffer, and water.
Hello. So now this is the next part of the PCR practical. So as I mentioned, these are the total 8 things that we need to add gradually, one after another, and we will set up one total PCR mixture, and we will prepare the PCR mixture in PCR tube, and we will put the PCR tube in the PCR system with a specific PCR reaction condition that I am going to tell you later. So we need the genomic DNA. So genomic DNA, we have got this genomic DNA isolated from the E. coli cells. We need to add 9 microliter. Forward primer that we have specific for that one gene of the E. coli DNA, that is 3 microliter. Reverse primer, we need 3 microliter. DNTPs, so that A, T, G, C and G, the DNTPs, it's a 10 millimolar, it's you have procured it, it's commercially available. So 10 millimolar DNTPs, we need to add 3 microliter. Taq enzyme, that is the Taq DNA polymerase, we need to add 1.5 microliter. Taq buffer, which is specific for this enzyme, this buffer, we need to add 15 microliter. MgCl2, we call it a PCR additive, it's a 50 millimolar of MgCl2 effective concentration, we need to add 4.5 microliter. And finally, we need to add the nucleus-free water with 111 microliter. Now, if you just add all this volume, you'll be getting that total volume is 150 microliter. Now, we have to distribute this total 150 microliter in three different PCR tubes with 50 microliter each. Okay, so the next part, how to fix the PCR, I am going to tell you in the next video. Okay. Hello. So the next part of the experiment. So first of all, we need one ice box which is full of ice, because we need to set one experiment by keeping all the component in ice. We need a different size or different volume capacity of the micropipettes. we need one small tray so that we can put different size of microgrids here. And in the ice box, we have kept all the reagents that I mentioned just earlier. So we have got the template DNA. So the template DNA that we want to use for the PCR reaction. We need two primer. One is the forward primer and reverse primer. So we have got the Taq DNA polymerase and the buffer which is used for the Taq DNA polymerase enzyme. We have got this nucleus-free water here. So it's a nucleus-free water. It's a double autoclaving. And we need one MgCl2 solution with a 50 millimolar strength. So just to increase the efficacy of the PCR reaction. Now, real time how to set one reaction using the PCR tube, I am just going to discuss this. Just wait. Hello guys. So we have come to the last part of our PCR practical. So as I described in the last video that I have prepared 3 PCR tubes. Each PCR tube contains 50 microliter of total PCR mixture, and as you can see that I have taken the PCR mixture in this ice box. So here I will going to set for the PCR reaction of these 3 tubes. Now this is called the thermal cycler. It is from the applied biosystem. Now I will just going to tell you that how to set the reaction parameters for one typical PCR reaction according to our genomic DNA and the primers that we have taken for our experiment. Okay, wait for some time. Okay, so whenever you put on the switch, and the main switch for this thermal cycler system, so initially it will show you a window like this. So here you have the setup the run. There is one sign in option, but we don't need any sign in option for that. Here we have the new method that you want to create. If you want to create any new method, so you can go to this button. So if you have already done some previous PCR experiments and you have already saved the PCR parameter, so here is the option for you, that is open method. So you can go to the open method, and there are some settings also, and just incubation. So at what temperature and what condition you want to incubate your sample, that is not for this PCR purpose. Rather, suppose if any experiment it says that okay, just incubate your sample 16 degrees Celsius for 8 hours. So you can just put on the lid, and you can see that there are some grooves. So you can put your your PCR tube, and you can just close the lid, and you can just incubate it at certain temperature or condition according to the requirement of your experiment. Okay, so let me show you. If I choose this option, that is open method, like this, so you will see that there are different PCR reactions that have already been done, and all the parameter, all the experiment, it is already been saved according to the last access or according to the setup date. So you can choose, suppose yesterday you did some PCR practical. So suppose if you saved this PG2M, so you will see that what are the basic parameters for the PCR reaction that you have set, like starting from the denaturation and final extension. Now we can anytime you can just you can edit your profile, you can edit your system parameter, or you can also cancel it, and you can go back to your main menu. Okay. Okay. So I am just going to select this setup run, and I want to create one new parameter for today's practical. So open template is the process. So once you just click the open template, so these are the things, it will automatically open. I will and you can see a kind of a window like this. So there are different sort of variety of the PCR that we can also go do for using this process. But initially for any practical, just we will select this basic PCR. Basic PCR, and in the right hand side that is basic PCR tag. Now once you just click this option, you can see entire parameter that you want to set up. This is a basic template. If you just find that okay, this template will serve the purpose for you, you can also use this, or anytime you can just edit this. So edit means like your stage 1 is the final initial denaturation, then your stage 2, that means your initial denaturation followed by the annealing, then extension, and finally you will be doing the final extension for certain period of time. And after the PCR, suppose you have done the PCR but you are doing some other work, you cannot go for some other post PCR experiment. So there is one option, that is you can see that 4 degrees Celsius for infinite period of time. So you can keep the system on for 4 degrees Celsius. Your PCR experiment will be stopped, but your sample will be incubated for your further use at that 4 degrees Celsius. Okay, so now there is a new file that I want to set up, a new experimental condition for today's PCR practical. So you can see that 95 degree is the temperature for the denaturation. So I have kept the total time for at 95 degrees Celsius for 1 minute 30 seconds. Then in the stage 2, the the denaturation is again 95 degrees Celsius for 30 seconds, followed by the annealing temperature that I have kept 50 degrees. So as I mentioned in my protocol, that this annealing temperature may vary depending on the DNA, that template DNA that you are going to use, the primers. So depending on the melting temperature or TM of the your primer, so you can choose this annealing temperature. So here 50 degree for 30 seconds, and 40x means total 40 cycle, 40 run will be done using this condition. Now the next part is the 72 degrees Celsius, that is the extension for 45 seconds, and in the stage 3, that is again the final extension at 72 degrees Celsius for 10 minutes, followed by the incubation, constant incubation at 4 degrees Celsius for certain period of time. Okay. Okay, so after setting the parameter, when you just finish the parameter setup and press the button next, so it will show a window like this. So here you can see there is a red the circle, it shows the ready. That means your parameter setup, your instrument is already, you have set in that those parameters that you want to use for your PCR experiment, and you can see that your parameter is set using the date for today, that is 13/9/2020. Now you can just open the lead here. You can open the lead here, and one by one, so you can have the PCR tube where you have taken the PCR sample. So I can just put the PCR tube here. So that means just if I show you closely, so this is how you could just want to fix, and then just close the lid, and then there is a button called start run. So you can now start your run. Now the window shows that your running has already started. So total time will be taken, that is 1 hour 56 minutes. So that will be the total time required for 1 hour 56 minutes. Anyway, So we will just open the lid after the experiment is over, after 2 hours, and then we will check for some downstream experiment also. Okay, so bye bye for today, guys.